Have you ever wondered what lives in a drop of water, or on your skin? These tiny worlds are teeming with life, invisible to the naked eye but crucial to our existence. Millions of tiny creatures called microbes are everywhere. They inhabit every corner of our planet, from the deepest oceans to the highest mountains. We can't see them without a microscope, but they're super important. These microorganisms play vital roles in ecosystems, human health, and even in the food we eat. Microbial culture is like creating a tiny neighborhood for these microbes in the lab. Scientists cultivate them to study their behaviors and interactions. We give them food, a comfy temperature, and everything they need to grow. This controlled environment allows us to observe their growth patterns and understand their needs. Then, we can study them up close. By examining these cultures, we gain insights into their functions and how they can benefit us. Think of it like making a sourdough starter. You nurture the yeast and in return, they help you bake delicious bread. You give the yeast a cozy place to live and they reward you with delicious bread. Microbes are similar. They thrive in the right conditions and can be incredibly useful. We provide for them and they help us make amazing discoveries. From antibiotics to biofuels, microbes have countless applications. It's like having a miniature zoo right on your lab bench. Each petri dish is a window into a microscopic world. We can learn so much about these tiny creatures and how they impact our world. Their influence is vast, shaping everything from our health to the environment. Microbial cultures are like secret agents in the world of science. They help us solve mysteries and develop life-saving solutions. For example, doctors use microbial cultures to identify the culprit behind an infection. By growing the bacteria from a patient's sample, they can figure out which antibiotic will knock it out. Scientists use microbial cultures to study everything from how bacteria break down pollutants to how different microbes interact with each other. It's like watching a tiny soap opera unfold under the microscope. And hold on to your hats because microbial cultures are even used to develop new medicines and vaccines. By studying how microbes grow and behave, we can create treatments for some of the world's toughest diseases. Just like you wouldn't want to live in a messy room, microbes have preferences too. They thrive in environments that suit their needs, and that's why we have to be very particular about how we grow them in the lab. That's why we use different types of food or culture media to grow them in the lab. Each type of media is like a special recipe designed to meet the dietary needs of different microbes. One popular option is nutrient agar, which is like a buffet for microbes. It's a general-purpose medium that supports the growth of a wide variety of organisms. It's packed with all the good stuff they need to thrive, like sugars, proteins, and yummy nutrients. This makes it an ideal choice for growing many different types of bacteria. Sometimes we want to be picky about who comes to our microbial party. We might only want to grow certain types of bacteria while inhibiting others. That's where selective media comes in. This type of media contains substances that favor the growth of specific microbes while suppressing others. It's like using a bouncer at a nightclub. Only certain types of microbes are allowed in. This helps us isolate and study specific bacteria more effectively. By controlling who gets in, we can focus on the microbes we're most interested in. And for those who like a bit of drama, there's differential media. This type of media contains indicators that cause different microbes to change color. It causes different microbes to change color, making it easy to tell them apart. This visual cue is incredibly helpful for identifying and differentiating between microbial species. It's like a costume party for microbes. Each one dresses up in a different color, making it a fun and effective way to study them. Now, how do we get those microbes onto the culture media? It's a fascinating process that requires precision and care. It's all about the streaking technique. This method allows us to isolate individual microbial colonies. It's like painting with microbes, but instead of brushes, we use loupes. These loupes help us transfer tiny amounts of microbes. The most common technique is the streak plate method. This method is essential for microbiologists. We start by sterilizing a wire loop and dipping it into our microbial sample. Sterilization is crucial to avoid contamination. Then we gently streak it across the surface of the agar plate, spreading the microbes out. This step requires a steady hand. As we continue streaking, the microbes become more and more spread out, eventually forming individual colonies. These colonies are vital for further study. It's like planting a field of tiny flowers. Remember, cleanliness is key. Each colony represents a single type of microbe. We use aseptic techniques throughout the process to prevent unwanted microbes from crashing our party. This ensures our results are accurate. Think of it like washing your hands before eating. We want to keep things clean and avoid contamination. A clean environment is essential for reliable experiments.